So today we have cell wall, extracellular matrix, and cell junctions. Kind of combine all the outer stuff all at once, and then next week we start our journey through the cell membrane, which is an entire chapter in and of itself. So super important, but today we are talking about these three things, starting with the cell wall. Definitionally, it is an extracellular structure extracellular structure of plant cells. So only plants have it, a couple bacteria, but we're not going to get super into that. But mainly plant cells, just an extracellular structure. I mean, it's a structure that the plant cell has that is outside of its, like, I want to say person, but cells aren't people. Anyway, extracellular, outside of the initial plasma membrane. That's what I want. Functions. It's got lots of them. Protection is our first and foremost. I'll grab my little cactus friend. This is Rick. This is my cactus. All right. So, sturdy, not necessarily super bendy. Protects against if I were to beat it, which I try not to. But anyway, protection. Also, maintaining shape. While our eukaryotic cells have our cytoskeleton that we talked about, and that like structure that lines the inside of the cell membrane to keep it from being just like a sack of cell parts, the cell wall helps maintain our shape like the cytoskeleton would. We also prevent excessive water uptake. Because plant cells have those big old vacuoles for our water, right? If we didn't have the cell wall, it would just kind of keep stretching and stretching until it busts. But that cell wall kind of provides some sort of like, hey, you're getting too big, don't take up any more water, kind of stuff. Allows the cells to kind of have that like solid structure that allows the cell to like moderate how much water it takes. Cell walls also hold plants up against gravity. So we have our bones that keep us standing, that fight the force of gravity, right? Our bones, our muscles. This guy doesn't have any bones or muscles. It stands up because the cell walls keep us straight. So, there's that. Any questions about cell wall function? All right. So, like I said, it's not all plants, or not only plants. Protists, fungi, and some unicellular eukaryotes also have cell walls. There are no multicellular eukaryotes with cell walls. If they are multicellular, odds are they have cytoskeletons in place to keep them in their general shape. Fungi, protists, and our single-celled eukaryotes can have cell walls. It's not just plants. There are two types of cell walls: primary cell walls and secondary cell walls. So we're going to start with our primary cell wall. Change colors on you. So, primary cell wall. The primary cell wall is secreted by young plants. So young plants are secreted by young plants. It is usually thin and flexible. Think about a little bean sprout, right? It's pretty easy to maneuver around. It doesn't necessarily need all that protection from the wind because it's not super tall. You can't get up there to catch the wind in the first place. So those first cell walls that are secreted by young plants are typically thin and flexible. Between primary walls of adjacent cells. So between primary walls of adjacent cells, so if we got a cell here and a cell here, we're talking about this space in here. It is called the middle lamina. This is a thin layer of polysaccharides. Ooh, there's only one L in polysaccharides. Called pectins. Have you ever like made jams or jellies at home? Yeah. The little the gelatin-esque substance you put in is called pectin. So pectin is used in a lot of canning and a lot of jam and jelly making, if that's your ish. So, and essentially what it does is it glues those cells together. So when we're talking about making our jams and jellies with it, it's what gives it its goopy kind of substance or texture. So a bunch of polysaccharides called pectins that glue those cells together. As cells mature, we have to have some more structure in there. We can't just rely on our primary cell walls. So there are two things a cell can do as it grows. The first one is secreting hardening substances into the primary wall. Hardening. So some of them just have primary walls. They get older, they need more structure. They put some more proteins and carbohydrates and all that jazz into that primary cell wall. It hardens up. We don't need to do anything else. others create a secondary cell wall or secondary cell wall. Our secondary cell wall is between the plasma membrane and the primary cell wall. So if this is our cell, and we've got our primary cell wall, and this was our plasma membrane, our secondary cell wall would be between the two. Because the cell's not going to build it outside of the primary wall, they're just going to put stuff into that space between the plasma membrane and the cell wall. Often, it is several laminated layers. So, like sedimentary rock, have it just like is striated in layers. So often it's several laminated layers. Often it is several laminated layers. It is strong and durable. That offers cells support. So like wood. Our trees have these secondary cell walls. That's how we get like tree bark. They need more support because they're taller, more structured. This guy likely only has primary cell walls. All those guys out there have secondary cell walls. That is cell walls. Any questions about those? No? Right. Now we are moving on to the extracellular matrix of animal cells. So, just animal cells, because plant cells have these cell walls that connect them. So, ingredients, i.e., what the extracellular matrix is made up of. Glycoproteins, and other carbohydrate containing molecules. Glycoproteins and other carbohydrate containing molecules. The most abundant of them is collagen. Collagen comes up a lot when people talk about skincare, because we want to fill in those gaps between cells in the skin, keep them locked tight so they can keep moisture trapped in. A lot of times skincare kind of boils down to collagen. Essentially, the collagen forms strong fibers outside of the cell. Collagen makes up 40% of all of the proteins in the human body. So if we took us and we sucked out all of the proteins that existed in our bodies, all of the proteins, all the enzymes that catalyze reactions, all of our structural proteins, all of our transport proteins, 40% of those proteins would be collagen because it is a vast network of just like fibers of proteins that connect all of our cells. It's everywhere. There are fibers, or they are fibers embedded in a network of proteoglycans. Embedded in a network of proteoglycans. Proteoglycans are small protein cores with many carbohydrate chains. In fact, a proteoglycan is 95% carbohydrate. We got a little baby protein and a bunch of fibers coming out of that. So, let's see. Some cells are attached to the extracellular matrix by glycoproteins called fibronectin. So, fibronectin is another protein. So far in our extracellular matrix, we have our collagen, our glycoproteins, or our proteoglycans, sorry. Collagen, proteoglycans, and now we have fibronectin. There should be an R in here. Fibronectin. They are glycoproteins. Now, glycoproteins just mean that they are part carbohydrate and part protein. So, fibronectin are also carbohydrate and protein things that attach cells to the extracellular matrix. And I'll show you a picture that kind of helps out. So, fibronectins and other extracellular matrix proteins bind to cell surface receptors called integrins. And those integrins are built into the plasma membrane with, they are connected to microfilaments on the inside. These transmit signals between the extracellular matrix and the cytoskeleton. Cytoskeleton, which helps regulate cell functions. So I'm gonna pull up a picture real quick because I feel like pictures help. So paste. All right, it's a little hazy, so we'll kind of doodle it next to it. So this is what our extracellular matrix looks like. So if we've got our plasma membrane, this is our plasma membrane. This is going to be our integrin. You guys want colored pencils? Okay. Our integrins. So our integrins are attached to microfilaments within the cell. So this is part of the cytoskeleton. These are our microfilaments. Now, on the outside, this yellow, try to keep the same color. This yellow is our collagen. So, collagen is a glycoprotein or carbohydrate containing molecule proteins. So, we've got our collagen. They just kind of float everywhere. I'll do another one just up through here. These guys are everywhere. That's our collagen. Wrapped around our collagens, we have our proteoglycan processes. So when we connect our proteins to our collagen, that is our fibronectin. So as we recall, our fibronectin, where to go? Binds to cell surface receptors and connects our extracellular matrix. So our fibronectin is what connects our integrins to our collagens. And then all throughout here, we have a bunch of proteoglycan. What's the word I'm looking for? Complexes. just other little fibers that help send signals and keep everything stuck together. So eventually, if we really thought about it, there would be another plasma membrane up through here, because it is the extracellular matrix, so between cells. We would have another integrin protein that is also connected by fibronectin, and our other microfilaments on the inside. Help a little bit? Ooh. So, main points here: integrins, fibronectins. The yellow is collagen. This helps regulate cell behavior. If one cell is experiencing super low drops in pH, or they're not getting enough energy, they can be like, yo, hey, there's something happening. We're not getting enough energy. And then the other cell can react to that and do whatever emergency preparedness stuff that it has to do in order to stay alive in low energy environments. All right. That is the extracellular matrix. We are now going to talk about cell junctions. Cell junctions. There are 3 animal cell ones and 1 plant cell one that we are going to talk about. Cell junctions. So, cells in both plant and animals are organized into tissues, which make up organs, which make up organ systems. Relatively easy concept, but essentially that means that we have to communicate with one another for these cells to work together in order to form tissues, in order to form organs, in order to form organ systems, because everybody's got to function. Neighboring cells often adhere, interact with, and communicate with cells via sites of direct contact. And those sites of direct contact are going to be our junctions. Yeah, so I had a little bitty paragraph about the cell ones, and then I made a chart for the animal cell ones. So in plant cells, actually I don't like that I used it. So plant cells, the main junction is called plasmodesmata. So plasmodesmata are perforations in cell walls. perforation in cell walls that connect cells. Allowing cytosol to pass through, joining their chemical environments. Creating one living continuum. So if this is a plant cell and this is a plant cell, this would be our plasmodesmata. So let's say something bad is happening in this cell. It will, the cell, whatever is wrong with it. So let's say, we'll just say that like we got an increase in pH, right? We have no clue where this increase in pH is coming from. So we've got an increase in pH. That increased pH can leak into the other cell and allow that cell time to react before it reaches the same fate. So if this cell dies, this cell can be like, uh-oh, pH is rising, let's do something. Let's release those buffers and lower it back down. Any questions about plasmodesmata? Okay. So, there are 3 types of animal cell junctions. They are tight junctions, desmosomes, and gap junctions. Ooh. First, we are going to talk structure. Let's see. Our plasma membranes are tightly bound together by specific proteins. So, let's see. Proteins tightly binding cells together. Sorry. We're just gonna do that and hope that's enough space. We also have our functions. Our functions, these act as a barrier for, so extracellular fluid doesn't leak across the layers, especially in epithelial cells. So barrier prevents extracellular fluid from leaking across layers. Usually this is epithelium. So, like our skin cells, our epithelium cells, they are sheets of cells. So in order to keep stuff from leaking from layer to layer, we've got these tight junctions. And the last one, we're going to draw it. So if this is a cell, and this is a cell, our tight junctions are just little proteins that hold them together. Next, we have desmosomes. Going all the way back up. Whoa. Desmosomes. They are intermediate keratin filaments. Or actually, sorry, nope. They are anchored by intermediate keratin filaments. Read that wrong. Anchored by intermediate keratin filaments. Functionally, they act as rivets that fasten cells together. In tight sheets. So if we've got our layer of skin cells, right, our desmosomes will act to lock those cells together, and those tight junctions act as that like second layer of defense to not let fluid leak through. So our drawing, this again is our cells. These are our keratin filaments. They are anchored in the cell, and those rivets kind of bond those things together. So, like that. One last one. They are gap junctions. Gap junctions are membrane proteins that surround pores. Membrane proteins. I'm out of space, so I'm going to abbreviate proteins. That surround pores in the plasma membrane. Where small molecules pass through. This is junction. All right. Functionally, they provide cytoplasmic channels from one cell or the other. So they essentially act like the plasma desmosomes did for the plant cells. So they act as cytoplasmic channels. desmata, sorry. Like plasmodesmata. Essentially, that diagram I drew for the plant cells, right, where it connects the cytoplasm of the two plant cells so they can act as one big organ system. The gap junctions do the exact same thing for animal cells. So if we've got our cells again, it would essentially be like one big protein that allows stuff to pass through. Here we go. So stuff can go from one cell whoop, to the other cell. Does anybody have any questions about cell junctions? I know that was a lot all at once. I do not believe there is an exit ticket, but I will check. That is semester 2. I don't want semester 2. There is not an exit ticket today. You are all good.